Welcome to the C-Suite Series presented by Channelchek and Noble Capital Markets. Noble is an SEC-registered FINRA-licensed broker-dealer and the source of the equity research available on Channelchek. Of the 6,000 small and microcap companies listed on the site, today we're featuring Lineage Cell Therapeutics. Ahu Demir, Noble's biotechnology research analyst covering the company, will be interviewing CEO Brian Culley and CFO Brandi Roberts. Afterwards, she will be joined by Michael Kopinski, Noble's Director of Research, to discuss the key takeaways. As a reminder, Ahu's research is available at no cost on Channelchek. Just type in Lineage Cell Therapeutics in the top left Company Data section. And now, here's Ahu, Brian, and Brandi.
Here is Brian Colley, the CEO of Lineage Therapeutics, and CFO Brandi Roberts joining us today. Thank you very much for joining us, and I would like to start with an introduction from both of you.
Well, thank you, Ahu. It's a pleasure to be here. So I'm Brian Culley. I'm the CEO of Lineage Cell Therapeutics. I've been in this position for approximately 2 years, and I have an extensive background in biotech product development, both on the scientific and business side, and just been really energized by working now in the last couple of years here at Lineage and focusing on cell therapy.
Great, thanks, Brian. And I'm Brandi Roberts. I'm the Chief Financial Officer at Lineage. I joined the company in January of 2019. This is the third company I've been CFO of. I started off my career as a CPA at PricewaterhouseCoopers and got my drug development training at Pfizer. Love working at small biotech companies, and I'm really excited about the cell therapy assets that we have.
Sounds great. Thank you very much. So I'll start with my first question. Since you joined Lineage 5 years ago, could you maybe provide us some history of the company and the changes you implemented, and maybe provide a quick overview of Lineage's current portfolio?
Well, thank you. Yeah, I think my hire 2 years ago was really to help the company focus down on its cell therapy assets. So cell therapy is still an industry which is in its infancy, and there were many things that the company was engaged in and pursuing, but I really felt strongly that the greatest potential for growth and share price appreciation was going to be around the cell therapy assets and advancing them through clinical programs and taking advantage of all of the discoveries of recent years and understanding how best to manufacture these cells and control them and scale them up and all the things that you need in order to have a real product at the other end that can get to patients. And so we made a lot of changes in how we manage the company, including even the management team, and really emphasized the directed differentiation of cells and transplanting them into the body to treat various diseases and conditions.
Sounds great, Brian. So prior to discussing every pipeline asset, I would like to maybe start with giving some information on Lineage's technology platform, the foundations of cell therapeutics that you're using, and also the manufacturing capabilities in general.
Yeah, cell therapy is a growing field, but it does have many different aspects. And so how Lineage applies cell therapy is what we do is we utilize the power of pluripotent stem cells. So these are cells that can be reproduced without any changes. So you can multiply them into enormous numbers. And then we run them through a process in order to direct their further differentiation into specific cell types. So these are cells that before we touch them, they're capable of becoming any of 200 different cell types in your body. So you can make them into bone cells or brain cells or liver cells or kidney cells. And what we do is we have those recipes. We know how to manufacture the specific cell types, and that really is our core technology, is having an easily accessible, right at the bench side, frozen vials of cells that can be transplanted into the body. And we have 3 clinical programs right now. And one of the key advantages of our company is the manufacturing of these cells. Scale is so important in order to keep costs low in cell therapy. And we have solutions that are applicable for essentially every patient. So they're not patient specific. These are sort of one size fits all applications. And we have already demonstrated an incredible ability to scale. When we run our retina cell program, for example, every time we run a batch, we're making about 2,500 administrations worth of material, clinical trial material that can be calculated from a 5 billion cell batch. So we are harnessing both the recipe to make specific cell types, transplant those into the body, as well as introducing some of the manufacturing features that are required to make sure that these products and programs have commercial viability.
And Ahu, I'll just add that, this is our facility in Israel. This is a subsidiary of Lineage, so we get to control this manufacturing process. And this is really our center of excellence. Our group in Israel are really experts at making these cells, and we've been really impressed with their capabilities as they've worked on Oprogen and now OPC1 as well.
Sounds great. Thank you, Brandi. So talking about the subsidiaries, I know based on your history, you had many subsidiaries. Now you actually made a lot of changes since you took over the management. So can we talk about how many subsidiaries you have and what are their capabilities?
Yeah, at one time, the company probably resembled more of a holding company. It held very large equity positions in subsidiary companies, which actually provided a lot of capital that we've used for our development. So we've benefited our shareholders by not having to do a traditional equities financing for nearly 3 years. Instead, we were able to spin off technology out of our massive patent estate, create new companies and new businesses, and allow those to grow and mature independently and create value for our shareholders in that way. At this time, the one subsidiary that's really meaningful is the one in Israel, the manufacturing facility, which we're 99% owners of that facility. So in terms, we do have some marketable securities. We have some positions in some other businesses which we can still access to help fund our primary operations. But we really have evolved the company over the last couple of years from the spin-out model into more of a traditional biotech, high-growth biotech company.
Yeah, that's very true. So Brian, you have currently 3 ongoing clinical trials, and could we maybe give some information, the names and indications you are going after before we dig into data and what the market opportunity might be?
Yeah, that makes a lot of sense. I described that we have the ability to manufacture particular cell types. So there are 3 of those programs which are in the clinic right now. So the first is that we have the ability to manufacture a specialized retina cell. So there's a condition called dry age-related macular degeneration, and the hallmark of that condition is that the death of retina cells in the back of the eye. So we manufacture brand new retina cells, and we can transplant them into the eye to replace the ones that are dying off. And in doing so, we're trying to preserve or even improve patients' vision. Is a massive opportunity for us because there are no approved therapies to treat dry age-related macular degeneration or dry AMD. So that's the first iteration. The second iteration of this technology is that we can manufacture a special kind of spinal cord cell. And we do that because some individuals, when they have a spinal cord injury, often from something like diving into shallow water or falling off of a mountain bike or something like that, the injury leads to cell death. And if you have cells of the spinal cord which have died off, you lose the ability to have a connection between your brain and your digits. You cannot move your upper extremities, so you're paralyzed. So what we do is we manufacture the cells that have been destroyed, and we transplant them into the body because we're trying to restore the connections, the wiring, and the insulation, which allows you to have motion by carrying the signals from your brain. There's nothing really available as an interventional therapeutic for patients who suffer from these spinal cord injuries. I'm sure we'll get into it. We've seen very exciting data from those patients as well. So that's the second iteration, is making spinal cord cells to treat spinal cord injury. And then the third and final iteration, which is currently in the clinic, is that we manufacture a different kind of cell called a dendritic cell. And dendritic cells are one of the most important cells in your immune system. They're responsible for providing information to your immune system, specifically to your T cells. So your body's T cells are the part of the immune system which is capable of destroying invaders, including a tumor. Tumor cells are essentially invaders. They become non-self. And so those T cells are prepared to be able to destroy tumors, but they need the information. They need to be able to recognize the tumor. And that information can be delivered by dendritic cells. So we manufacture dendritic cells and we load them with information, and then we inject them into the patient. And those millions of dendritic cells go around the body and they provide that information to your T cells, so the T cells know what to destroy in terms of foreign invaders. So it's 3 different programs. It's a vision program, it's a spinal cord injury program, and it's an oncology program. And each of those, again, are in the clinic right now with each of those different cell types.
Great overview. Thanks, Brian. So let's start with OpRegen. So can we talk about the data you have gathered up to date, and also the market potential? As we know, there are no approved treatments on the dry age-related macular edema. And what are the, however, the developmental landscape is quite crowded. So if we could elaborate on the differentiating factors and how you would fit into the landscape.
Yeah, I think this is core to understanding why we think cell therapy can be successful in this setting. So as I mentioned earlier, the hallmark of dry AMD is the death of these retina cells in the back of the eye. And it affects, in a more serious form, approximately 2 million Americans, but there are no approved therapies. So everyone recognizes and agrees that this is a multi-billion-dollar commercial opportunity. But it's a head scratcher because we don't understand why there aren't approved therapies. Why has it been so difficult to treat this condition? Our view of it is that it is an advanced condition. By the time your cells are dying, there's so many things going wrong that if you try to treat that problem with a small molecule or an antibody that only hits one pathway or one target, there are probably 400 other things that are going wrong that fixing that one target is probably not enough. And there have been some fairly notable failures in the field, probably attributable to people who are trying to modulate one pathway in the cell and not being able to do anything about all the other pathways. So our approach is different. We replace the entire cell. We give you a brand new retina cell. And in doing so, if there are 400 things which are broken with those cells, we're replacing all 400 things. My favorite analogy is that if I need to go somewhere in my car and it doesn't start, I don't really care if it's the battery or if there's no gas. I just need a new car so that I can go and make my trip. That's the same analogy here. We manufacture brand new retina cells. Now, in doing so, we think that this is a very nice application of cell therapy where it can be successful where other approaches have failed. And we're starting to see some of that success in the clinic. We've treated approximately 20 patients to date, and we've broken them into 2 large groups. So the first 12 patients that were treated, they were all legally blind. So these are people who have got very advanced disease and perhaps too advanced to really see a big benefit. But it's important from a safety perspective to start with patients who are legally blind. And in doing so, we saw some very encouraging results. We saw that our cells were durable. They remained for a very long time, for multiple years. The cells were well tolerated, which is important safety consideration. We saw that the surgical procedure was fairly straightforward. It only takes about 20 to 30 minutes. So it's kind of about how long it would take for a glaucoma surgery or something like that. So in those first 12 patients, we gained a lot of information. And that allowed us to move into the next group of patients where we're going to do another 10 or 12 patients. And these are individuals who have less advanced disease. So they're not quite so far gone. They're earlier in their disease. They're earlier with their condition. And they have better baseline vision. And some of these individuals can still drive cars. So they've got something to work with in terms of being able to treat their vision. And we have administered our cells called OpRegen cells. Those retina cells have been administered to patients And we've seen some wonderful benefits. Some of the patients have had substantial increases in their vision. So we had someone who could read for a durable time for approximately a year now, have been able to read between 20 and 25 letters more on an eye chart, traditional eye chart. So that's 4 or 5 lines lower. So really significant differences that are difficult to imagine are due to chance. You know, if someone improves by 20 letters on an eye chart, you kind of think it was probably because of what you did in transplanting the cells into their eye that provided that benefit. But it's not the only patient that we saw those kinds of changes. We saw a patient who had a doubling of their reading speed. We've seen organizational and structural changes to the retina. So the nice thing about the eye is you can look inside of the eye and you can image a lot of things that are going on there in the eye. And many of the patients in this better starting cohort have benefited. They've seen increases from as little as 4 or 5 letters to, as I say, upwards of north of 20 letters. And so we're really excited because we're now finally operating in a group of patients that really are resembling who we expect that this treatment is designed for. And because they have better baseline vision, we can see those changes over time. And then, completely unexpectedly, we saw something that is called retinal restoration. And I really, this is such a profound change that we're so excited about seeing this in this patient, that if you think about the wound, right, in the back of the eye, these retina cells are dying off. They roughly die off in a circular pattern, and that circular pattern gets larger and larger over time, and it expands outward as more and more cells die off. And every company, all of us, have really been approaching this disease and thinking about how could we slow that growth? How can we make that area of atrophy grow slower? Because you're saving vision if you can do that. But we had a patient recently who actually ended up with their area of atrophy that at 9 months had gotten smaller. That's incredible. I mean, this is exactly what the promise of cell therapy was designed to do, is to be able to replace lost function. It's almost like we're replacing little pieces of the organ. Think of the retina as an organ in and of itself. So this is very exciting. We need to reproduce this. We want to show that we can do it more than just once, and we're working on treating more patients so that we can show it. But I think what we're seeing with cell therapy is we may be rewriting what is thought to be possible in the setting of dry AMD by demonstrating with a lot of convincing evidence that we were able to actually shrink the area of atrophy, which could have very profound benefits for this patient, who, by the way, is also seeing better.
Sounds great.
Yeah. And I was just going to add, I mean, this market opportunity is really amazing. You know, we talked about the fact that there's no dry AMD treatments available right now. There are wet AMD treatments, though, and we know that they make over $10 billion a year. And, you know, wet AMD is a lot less prevalent than dry AMD. Dry AMD patients make up 85 to 90% of all AMD. So, you know, this is a really exciting opportunity. You mentioned it is a crowded field there. We do have competitors, but we really do think that OpRegen can be a very good treatment option. Especially when you look at others who are thinking about monthly injections as the wet AMD patients get currently. You know, we're thinking that this might be something that you only need to get once, or maybe even every couple of years. And so that is definitely a game changer when you think about OpRegen and what we might be able to bring to the market.
Brandi, actually, that's a very good point, because that brings me to my next question. As we know, treating eye is not very easy in terms of the administration. So I wanted to ask about the Orbit device and how you actually overcome that hurdle a little bit, and considering the dosing in terms of OpRegen administration.
Yeah, we have entered into an exclusive option agreement with a company called Gyroscope, and Gyroscope received 510(k) approval for a surgical implantation transplant device. And so they have let's call it special needle and a method for delivering cells to the sub-retinal space. So that's the destination. That's where we're trying to put our cells. Our cells are a suspension, so they're floating around in the medium, but they need to get delivered to a special area. And once they get there, they settle down into a monolayer, and they can begin performing the functions of retina cells. So there is a traditional way of administering these cells, and then there's the way that we're approaching it. So the traditional way requires you to remove the vitreous. You put a needle through the front of the eye, and you actually puncture the retina. So you have to get underneath the retina. And then once you have arrived there with the end of the needle, you then discharge the payload. The cells get administered underneath the retina. So you can imagine if I were trying to, put a needle just between some layers of skin and deliver the cells, and I would form like a small little blister or a bleb on the top of my hand. It's the same kind of scenario with the retina. However, the problem with that traditional method is that when you remove the needle, there is an efflux of cells. They can come out of the hole that you punctured in the retina, and then they can float around and they can land in funny places and they can begin to divide because they are living cells. And so that can cause some complications that may even require a second surgical intervention. So we entered into an agreement to evaluate the Orbit subretinal delivery system. And what this device does is a very clever design. It comes around the back of the eye and up underneath the retina. So you never take out the vitreous and you never puncture the retina. It's almost as if the needle came up magically underneath the skin, and you're able to deliver the cells to the subretinal space. And then when you remove the needle and the whole flexible cannula comes out from behind the eye, you have not punctured the retina, so there's no hole there for the cells to go into the vitreal space. They stay in the subretinal area. There's a bit of positive pressure in that space. And the beautiful thing about that solution is that if you don't lose cells to efflux, it means you have perfect dose control. And dose control is so important because dose control means that you have less variability in your clinical trials. Less variability means smaller, faster, cheaper clinical trials. It also, using this device, may mean that we see fewer side effects called epiretinal membranes. And in fact, we have not seen formation of epiretinal membranes when using this device. So we haven't made a decision for sure, but what's really nice about it is that the device holds a lot of promise. The surgical procedure is very comparable to what people are doing in the standard way. It's just a half day of training, and you're ready to go. You're qualified to use this device. The surgeons have given us feedback that they really like it, and we're the only cell therapy company that has the right to use it in this disease. So even if you're willing to accept that other approaches to treating dry AMD may be challenged by hitting just a single target, and that cell therapy is the way to go. Even within that group of cell therapy companies, we're the only ones that have the advantage of being able to use this device for this disease.
That's a big advantage, I would say, Brian. That's great hurdle to overcome. So moving into OPC1, the second asset, can you tell us about spinal cord injury and the data you gather for OPC1 in this indication and the potential market opportunity / competitive landscape?
Yeah, so just as a reminder, we're talking about the spinal cord itself, right, within the bones of the spine, and that it can become injured, a contusion injury, and that can lead to inflammation, and that's a damaging and hostile environment for cells. And so there are these special cells called oligodendrocyte progenitor cells. It's a form of glial cell that can die off. So when those die off, there's literally a hole, a cavity, a hole that is formed. Well, if you have a connection that's supposed to run from here to over here, and now there's a cavity, there's a gap, that electrical impulse cannot jump across the gap. Electrical impulses can't leap that way. So you need some material to fill that cavity. So we put oligodendrocyte progenitor cells into that space. These are the cells that can manufacture myelin, which is the insulation for the axons. So it's not just that you need to grow the axons, they need to grow, and they need to myelinate, and they need to fire at the right time. So there's a lot of things that are going on in the spinal cord after injury. And so what we do is we manufacture away from the body we are able to manufacture these oligodendrocyte cells, and then we put them into this this cavity. And what we're looking to do is we come in about 3 to 6 weeks after the injury. So this isn't like a stroke where you have to be there right away. We're showing up weeks after some of the more severe inflammation has calmed down, and we're putting our cells in and we're filling that cavity. I jokingly call it, it's like spackle for your spinal cord. And then what we're hoping is that the axons on either side of that space will begin to innervate and connect and be myelinated. And that in combination with all the other things that these patients are accustomed to doing in terms of their recovery, their physical therapy and all of those things, we don't require you to skip any of that. That's all important. We want patients to do that. And what we're really trying to do is provide patients with the substrate or the material which will give them more mobility after an injury. So the difference between being able to feed yourself, to be able to type instead of not being able to type. I mean, these are massive changes, and it can be really subtle. I talked to someone who had suffered a spinal cord injury. I spoke with him a couple of weeks ago. He's a few years after he participated on a clinical trial. He's doing really well. He's a student in college now, and he told me that just being able to waggle his finger gave him enough control to be able to help him feed himself. So even little changes can be beneficial. But we've seen great changes. We've seen really significant changes. We've treated 25 patients so far. Let's see, 96% of them had a successful engraftment. So the cells have gone in. They didn't grow into funny things. I mean, the cells are sitting there, the resident. They're not causing complications. They're not causing adverse events for the patients. They're very well tolerated. And about a third of the patients that we treated had what we call 2 motor levels of improvement in their upper extremities. So if you want to think about, like, what does a motor level mean? What does it mean to an individual to gain 2 levels? Well, at a C4, right, cervical level 4 for a level of injury, that kind of injury would require 18 to 24 hours of care, support around the home, helping people feed, dress, all the normal activities of daily living. So that's a C4. Well, if you can move them 2 levels to C6, well, now you're talking about just having 4 to 6 hours of help around the house. So instead of almost around the clock support and care, you're talking about, you know, I can't reach the upper levels of the pantry kind of things, you know, or maybe, you know, you need help with if you've got a stacked washer dryer and you know you can't reach the upper unit. It's life changing to go 2 levels, and fully 32% of our patients gained 2 levels in mobility. And we think compared to what you'd expect normally that's a big improvement and a big change. And so that's what we want to be able to do, is we want to provide these massive quality of life improvements, give people more flexibility and mobility, because I have to say, typically it's very young individuals, it's young males, prime of life stories, 16, 18, 20 years old, who most frequently end up with these injuries. And so we're trying to give them the ability to drive their car and you know, go to college and all those sorts of things.
Well, and I think, again, when you talk about the market opportunity for something like this, you know, there are about 18,000 spinal cord injuries a year in the United States. Studies show that lifetime costs are about $5 million for every spinal cord injury patient. So if we're able to do what Brian talked about and move somebody from 18 to 24 hours of care to something like 4 to 6 hours of care, we're able to make a big change in that level of care that's needed and that cost that will be over a lifetime, because as he said, these are young kids. They've got their whole lives ahead of them. And so if we're able to provide them to have the movement to be able to type, that means they could probably have a job. As we talked about, there is a patient who's in college right now. You know, we want them to be able to go forward and be able to have incomes and move forward with their lives. And so we think that you know, that presents just an amazing market opportunity in that indication as well.
That's great. That sounds really helpful for, I think, for our audience. They have a much better idea. So moving into the active program, you recently disclosed data from this program for the first time. So would you be able to provide some color on the data and also your partnership with Cancer Research UK and your plans for this program?
Yeah, the VAC program is our third program. So again, we're manufacturing dendritic cells to help educate the T cells of your immune system to fight tumors. And we did. We announced very recently the first data from this program in non-small cell lung cancer. So we've treated just a small number of patients so far, 6 patients. But what we were looking to see from this clinical study was we wanted to see evidence that the information which we put into the dendritic cells, that it reached the patient's immune system, and that we could measure and identify specific and targeted reactivity to our antigen by the patient's immune system. And that's what we were able to report. And that's very exciting to us because that is the proof of principle. That's the mechanistic principle that we're looking for, that we can put our message of choice. We decide what the information is. We put that in the dendritic cell. We get that into the patient, and then that patient's immune system picks up and amplifies that message. So we're very happy not only that that message was detected, but it's detected at very high levels. We saw immune cell reactivity north of 2%, north of 3% even in some cases. I think the highest recorded amount was 3.8 or 3.6%. So very high levels of reactivity that were engendered following our treatment. So that was very exciting from a mechanistic standpoint. This study, as you just mentioned, is being run by a partner of ours, Cancer Research UK, largest cancer charity dedicated to saving lives through cancer research. They're based in the UK. So the sites, the clinical trial sites that we have, there are 2 of them, one in Birmingham, one in Southampton. Those are both located in the UK. And Cancer Research UK is going to continue enrolling and treating patients, but we actually have taken a priority role. We've exercised an option to take control of the program back from Cancer Research UK because of the data that we saw. So the evidence of that mechanistic data that excited us, that was enough for us to say, okay, Cancer Research UK, we want you to keep doing what you're doing. Great job, keep it up. But we're going to also bring the program in-house, and we're going to send it over to our Israeli manufacturing facility. And what we're looking to do there is we're looking to mature and modernize some of the manufacturing. So a lot of what we did with the retina program, and we're in the midst of doing with the spinal cord program, we want to also do with the dendritic cell program. Investors or folks of all walks of life often take the manufacturing side of cell therapy for granted, but it's so critical to get it right. Oftentimes in cell therapy or in any new technology, you see the clinical data runs a little bit faster than the manufacturing. Well, you might have clinical data that's incredible, but if it costs, $3 million to treat someone, it's not really a product, is it? So you really have to find a way to drive down your costs of manufacturing and be able to get treatments that are accessible to everyone. And so we do that with our allogeneic approaches. Sometimes we look at autologous data as evidence and information to help guide us, but ultimately we want to develop allogeneic, meaning off-the-shelf approaches in both dry AMD, spinal cord injury, and all sorts of different types of cancer, because we can pick different messages and then we would look to treat different types of cancer. So it's really, we have a whole platform within this one program that resides within Lineage.
Yeah, that's a good point, Brian. Also, can we talk about what indication you are going after? As far as I know, the data was from lung cancer, which is a multi-billion-dollar market as well. Crowded space, but even a small percentage of market share would be very meaningful for Lineage. So what are the plans moving forward in the lung cancer or any other indication for VAC2?
Well, we think that the best way to use a dendritic cell vaccine would be in combination perhaps with checkpoint inhibitors or perhaps even with chemotherapy. And we haven't made a final decision there, and we haven't made a final decision on which indication. We may remain in non-small cell lung cancer. We may evolve or expand into other tumors with the VAC2 program. We need to collect a little bit more data, analyze it over time, and really look at the competitive landscape. But the biological rationale for a combination therapy is that the dendritic cell vaccine is, as I described, a good tool for educating the immune system. So you can think of it as like an accelerator. We're trying to accelerate the immune response to a tumor. Well, checkpoint inhibitors, they can be thought of as taking your foot off of the brake. Right? This is really, they're built on the brake technology. And so if you could use a combination where you're both knocking down the tumor's defenses and the tumor microenvironment, and that's for taking off the brake. And at the same time, you're using our dendritic cell vaccine, you're hitting the accelerator. We think that's where you might be able to get the most profound benefits in the clinic. And really, that seems to be the direction that the data is going in the field. Years ago, there was a product that was approved that was a dendritic cell vaccine. It was not off the shelf. It was patient specific. The economics were a little bit troubled, but the clinical data, did teach us a lot of things. And various academic and even some other biotech companies have been looking at the potential for dendritic cell vaccines. There's sort of a reemergence going on, and so we want to be in a leadership position in that reemergence and making really smart decisions about which tumor types to go into based on the antigen, based on the competitive landscape. These are all announcements that investors can look forward to going forward from us.
That sounds great. So does it mean using various antigens, you can actually apply RAC2 as a platform technology and apply into various indications? So it would be fair to say that.
Yeah, I think that's ultimately the direction that program needs to go. I prophetically talk about a VAC3, VAC4, VAC5. Perhaps there's artificial intelligence generated antigens. Some people are using combination antigens of neoantigens, tumor associated antigens. There are literally billions of antigens that one could choose from. And so there's this amazing project where you need to go through and decide, okay, what little piece of information am I going to put in the dendritic cell? Lineage provides the dendritic cell. It's the dumb carrier, but the real key is the piece of information that goes inside of it. So I think this gives us a lot of opportunities for partnerships with different entities who might bring the antigen and want to load it into our delivery platform. I mean, in theory, we could have a dozen programs in oncology someday. So yeah, you're exactly right that just picking and choosing different antigens, specifically targeting different tumor types, could open up a whole bunch of different opportunities down the road.
Certainly, yeah, I can for sure see that. Upon, I guess, meaningful data with starting indications, then moving to the various ones. Brandi, I will refer my next question to you. Could you provide us some information on the balance sheet? What is your cash position and cash burn rates currently?
Sure, absolutely. So our cash and marketable securities at the end of June were about $20 million. And we recently just added another $25 million to our accounts due to a note payment from Juvenescence related to a sale of Ajax shares that we did a few years ago. So we were really in good shape from our cash position, and we've worked really hard over the last couple of years to bring our burn down. As a reminder, these 3 clinical programs came together through a merger with Asterias. And so if you look at Lineage and Asterias in 2018, when they were separate companies, our cash burn was something like $43 million. But if you look at the first half of 2020, we were at less than $10 million. So we've worked really hard to find synergies, consolidate our manufacturing all in Israel. We made significant headcount reductions, really wanted to have a lean team in place, and we continue to look for additional efficiencies, but very proud of where what we've been able to accomplish. We've made significant progress with still these three clinical stage programs, and they all have billion-dollar market potential, and so really exciting place to be at right now.
That sounds great. And my next and last question would be if you could highlight some of the key catalysts, value-generating key catalysts in the next 6 to 12 months.
Oh yeah, that's great for us. I mean, just a few weeks from now is the AAO conference, so that's a major medical conference for retina specialists, and we're going to have an update on our data that'll be presented by a surgeon outside of the company, and so he will walk through and provide an update on enrollment and efficacy and safety and the use of the Orbit device. So November 14th to 16th, the AAO conference is the next data update on OpRegen. We have said many times before year end, we want to complete enrollment in the OpRegen clinical trial for dry AMD. That'll be a major event for us. We have said that we intend to provide an update on the spinal cord manufacturing. We didn't get into that a whole lot, but the old way of manufacturing the cells has been changed and improved, and we want to be able to provide some update and some detail around what we've been working on, because it's been kind of behind the scenes, and I think there's some surprise achievement that we've been able to accomplish over the last year. And then we still have a couple of patients that we expect Cancer Research UK to enroll in the VAC program. So, all of the programs have some important major milestones ahead, some of them clinical, some of them manufacturing, but definitely a busy time for the company, and we'll continue to push that news out the door so that people can follow the story and see what we're up to.
Sounds great. Brian, Brandi, thank you very much for your time, and we really appreciate all the information you provided. It sounds like a very exciting time for you in the next 6 to 12 months, and I'm quite intrigued by all the developments you are taking under your management. So we look forward to all the catalysts, and we'll be following the story. Thank you for your time.
Wonderful. Thanks, Ahu.
Ahu, this was extremely informative, and I thought that the management team gave really good explanations in detail. I learned a lot, but I was just wondering, in terms of the trial size, it seems pretty small. Am I wrong about that?
No, you're totally right, Michael. So the reason for that is the indications they are going after. Spinal cord injuries, really small indication, and also dry AMD. It's a large population. However, just the clinical trials run in much smaller patient populations, especially they're still early stage. They're in phase 1-2 stages. So that's generally very indication specific. Once VAC2 is conducted, the clinical trial, there will be a much larger patient population, as it's in lung cancer, and cancer oncology indications tend to run much larger patient enrollments.
Gotcha. Ahu, it seems that the company pulling their trials, or not actually pulling their trials, but shifting their focus to manufacturing is a little odd, but of course this company has manufacturing capability, which many might not have. Do you find that unusual?
No, it's not, Michael. So prior to Brandi and Brian joining Lineage, they ran as a holdings company, and now they are much more focused on the cell therapeutic side. And the manufacturing comes because they are doing off-the-shelf cell therapeutics manufacturing, which they have to do to run their clinical trials. So they do have this subsidiary. They only do the manufacturing side, and they're only focused on cell therapeutics in various indications. So that's their current focus, and it's not very unusual.
And he indicated that there were a couple of catalysts that are coming up that are good potential catalysts for the stock. I was just wondering, do you view those as key catalysts for the stock, and are there others that we should look for?
I certainly do. So the data they will be presenting on November, AO is a major value generating catalyst for their lead asset Oprogen. Back to clinical trials, that's expected next year, is also a major catalyst for the stock, because it's such a large indication. The moment they get into the clinic would be quite impactful for the stock.
Well, thank you, Ahu. It was a very interesting company. Thanks for bringing them to us.
Thank you. Thanks, Michael.
This has been the C-Suite Interview Series, brought to you by Channelchek and Noble Capital Markets. Log in to Channelchek or subscribe to our YouTube channel to get the full catalog of C-Suite interviews and virtual roadshow channelcasts. Register for Channelchek's no-cost premium content and receive notifications of new releases from both series. Thank you for joining us.